The human nervous system comprises many billions of neurons, all intricately interconnected to process a constant stream of internal and environmental inputs. To master this complex network for medical boards, we have to deconstruct it into its foundational histological blocks: neurons, the supporting glial matrix, connective tissue sheaths, and the specific pathologies that break them down. Anatomically, the system is split in two. The central nervous system houses the brain and spinal cord, while the peripheral nervous system consists of the cranial, spinal, and peripheral nerves, along with the ganglia located outside the CNS. Diagnosing clinical conditions like multiple sclerosis or rabies requires you to first understand the precise physical layout of these normal tissues. Understanding this physical architecture reveals how these systems function and exactly where they break down. The typical neuron is highly polarized. It has distinct geographic zones dedicated to specialized functions. The cell body, or soma, contains a large euchromatic nucleus. It acts as the trophic center, serving as the metabolic and synthetic factory for the entire cell. This micrograph and diagram show the soma's internal layout. Darkly stained regions are the chromatophilic Nissl substance, dense masses of rough ER driving protein synthesis. Notice the axon hillock, the pyramid-shaped origin of the axon. Nissl substance populates the soma, but is entirely absent here. Extending from the soma are the short, highly branched dendrites, specialized to receive stimuli. In contrast, a single constant diameter axon carries the generated nerve impulse away from the cell body. This structural polarization allows the neuron to collect and compute thousands of incoming variables at the dendrites, but execute only one binary decision down the axon: to fire or not to fire. Because the axoplasm lacks its own synthetic machinery like polysomes or rough ER, the axon relies entirely on an internal microtubule railway system for survival. Organelles and macromolecules move outward to the synaptic terminals via anterograde transport, driven along microtubules by the motor protein kinesin. Retrograde transport runs opposite. The motor protein dynein returns recycled materials and environmental samples from the periphery back to the cell body. Back on the receiving end, the small dendritic branches are covered in dynamic membrane protrusions called dendritic spines. These serve as the initial processing sites for synaptic signals and neuroplasticity. We categorize neurons structurally by the number of processes extending from the soma: multipolar, bipolar, unipolar or pseudounipolar, and anaxonic. The anaxonic configuration is found only in certain CNS interneurons. These cells have extensive branching dendrites, but lack a true axon. They do not produce action potentials, but instead regulate local electrical changes in adjacent neurons. The necessity of long-distance internal transport creates a critical metabolic vulnerability. The axon's reliance on the soma forms a fragile supply chain, easily exploited by disease. Neurons are supported by the neuroglia, a microscopic matrix that dictates the survival, insulation, and immunity of the nervous system. In the CNS, the oligodendrocyte is the primary myelinator. It extends multiple sheet-like processes, wrapping compacted lipid sheets around several nearby axon segments to speed up electrical conduction. Astrocytes act as master controllers. They use their perivascular feet to construct the blood-brain barrier and regulate the interstitial fluid composition. These terminal processes of astrocytes completely encase capillaries. This physical barrier facilitates transport while maintaining the strict environmental controls of the brain. Microglia operate as resident immune macrophages of the CNS. Unlike glia originating from the neural tube, microglia derive from embryonic yolk sac monocytes. Ependymal cells form a ciliated, epithelial-like lining along the brain ventricles and the central canal, actively assisting in the circulation of cerebrospinal fluid. The CNS lacks traditional collagen-heavy connective tissue. This complex glial web acts as both the physical scaffold and the localized defense matrix for the brain and spinal cord. The peripheral nervous system relies on an entirely different set of support cells. Unlike the multi-branching oligodendrocyte of the CNS, a peripheral Schwann cell, or neurolemmatocyte, dedicates itself to wrapping only a single segment of one axon. Within peripheral ganglia, satellite cells form a protective, insulating ring specifically surrounding the large neuronal cell bodies to regulate their microenvironment. Peripheral axons are packaged for protection. The entire nerve is bundled by the epineurium, a dense outer fibrous coat. Inside, the perineurium tightly groups nerve fibers into individual fascicles, establishing a distinct blood-nerve barrier. The deepest layer is the endoneurium, a loose connective tissue that intimately wraps each individual myelinated axon. This tough, multi-layered cabling provides peripheral nerves with physical resilience and regenerative capabilities, entirely missing from the central nervous system. The traveling electrical impulse eventually reaches a dead end at the terminal bouton, the dilated tip of the small axonal branch. The synapse has three geographic components: the presynaptic vesicle release zone, the physical gap of the synaptic cleft, and the postsynaptic receptor bed. These panels detail three anatomical configurations. Branched axon terminals connect to the soma, forming an axosomatic synapse, or to a dendritic spin, forming an axodendritic synapse. The third, the axo-axonic synapse, forms between two axon terminals, uniquely functioning to modulate the activity of the others. This histological junction is the site where the neuron's physical structure converts a traveling electrical impulse into a chemical signal. Board examiners test pathology by asking you to identify which normal histological block has been broken. Multiple sclerosis is an autoimmune destruction specifically targeting the oligodendrocytes of the central nervous system. In contrast, Guillain-Barré syndrome is an acute inflammatory demyelination, targeting only the Schwann cells in the peripheral nervous system. The rabies virus exploits internal transport. It enters through a peripheral wound, and it hijacks the retrograde dyning motors to travel all the way up the axon to the brain. In Alzheimer's disease, the histological markers are location-specific. Neurofibrillary tau tangles accumulate inside the soma, while beta-amyloid neuritic plaques form outside the cell. If an axon is severed from its cell body, the distal segment quickly breaks down in a process called Wallerian degeneration. If Schwann cells fail to properly align and guide the regenerating axon, the nerve fibers grow into a disorganized, painful swelling known as a neuroma. From autoimmune attacks to viral hijacking, neurological disease is a failure of specific building blocks. Recognizing the normal histology allows us to see exactly where the system has been compromised.